Hi students, this is a review of chapter 22 or the reproductive system. This is a chapter that is usually done over 2 or 3 lectures, so I'd recommend kind of breaking it down into the meiotic division, and then the male system, and then the female system. So if you end up kind of listening to the whole lecture, at once, it might be a little bit too much to kind of take in. So again, you want to kind of break it down into those 3 portions. So we'll start off with an introduction about the system, that we have 2 systems. There's a female and a male reproductive system that are connected through different organs and different glands, and there are lots of hormones that play into this. The hormones start off at in the hypothalamus, and then the hypothalamus would make gonadotropin releasing hormones that controls or stimulates the anterior pituitary to make a set of hormones as well, and then which are namely FSH and LH, and whether male or female, both have that set of hormones. And then FSH and LH are going to go to the gonads. So now we would differ whether it's a testis or an ovary and stimulate those gonads to produce their own hormones. The hormones are responsible for development and maintaining the secondary sexual characteristics. The reproductive organs are also responsible for making sex cells in order to transport them for fertilization. So we'll talk about meiosis. Well, meiosis is that kind of division, which is very different than the mitotic division. In mitosis, we are making what are known as somatic cells. So somatic cells are the cells anywhere in the body except, excluding the sex cells. The sex cells are the sperms and eggs or the oocytes. So any other cell in the body, hair cell, intestinal cell, nail cell, any other cell, think of it, you name it, that is known as a somatic cell. When you make copies of a somatic cell, that is known as mitotic division. Okay, that's not the process we're going to be discussing right now. What we are discussing is meiotic division, and that is the formation of these sex cells, where you'll take that cell of 46 chromosomes, and you have to split them into 2. So you'll end up making cells that are haploid, meaning that they have half of the chromosomal number. So you'll end up making 23 chromosomes, so that if fertilization occurs, you'd have a sperm with 23 chromosomes uniting with an egg that has 23 chromosomes, ending up making a zygote that now has 46 chromosomes again. So we've restored the 46 chromosomes all over again. So the aim of meiotic division is actually to split the number of chromosomes into half. Okay? And that is, again, your whole process of meiosis. Meiosis takes place over it's a series of 2 divisions. The first division is known as meiosis 1, and then it's followed by meiosis 2. In meiosis 1, what we're basically doing is taking that chromosome, and remember the X-shaped chromosome were made out of the 2 chromatids, and they are connected at the center by a centromere. So in the first meiotic division, the aim of the first meiotic division is to split those the 46 chromosomes into 23 chromosomes. Each of them has 2 chromatids. So I'll show you the picture to kind of explain that better. So this is the first cell we're going to start off with, which is 46 chromosomes. In your first meiotic division, you'll have all of these chromosomes kind of lined up in the midline, and you see that each chromosome is made out of 2 chromatids, and they are connected at the centromere. In your first meiotic division, you're going to end up having 23 chromosomes. So these 2 cells are haploid cells, meaning that they have 23 chromosomes, but each chromosome is still made out of 2 chromatids. So in your first meiotic division, you're splitting the chromosomes. So now you have 23 with 2 chromatids. In your second meiotic division, you're going to now split that 1 chromosome into 2. So you'll end up making these haploid cells, which again are 23 chromosomes, But each one now has just one chromatid instead of looking at like that X-shaped chromosome with two chromatids. So to kind of wrap up the differences between meiosis 1 and meiosis 2, in the first meiotic division, your aim is to split these chromosomes in half. You end up making haploid cells with two chromatids each in each chromosome. In your second meiotic division, your aim is to split now these chromatids from each other, and you'll end up making haploid cells where each chromosome is made out of just one chromatid. Okay, so that again, this is what we just talked about in writing, kind of, you can go over that, and if you have any questions about them, please let me know. The stages of each meiotic division are basically the same stages of mitosis. So you start off with prophase, and then metaphase, anaphase, and telophase. If you're in the first meiotic division, you'll give it 1, so prophase 1, metaphase 1, anaphase 1, and telophase 1. In your second meiotic division, you'll just name them 2. So there'll be prophase 2, metaphase 2, and so on. In prophase 1, this is where a lot of action is happening. And that involves synapses, which is basically the pairing of homologous chromosomes. So homologous chromosomes means that these two chromosomes are carrying the same characteristics. Okay, they code for basically the same genes. And when they come together in the midline and they are paired, the scientific term for that is synapses. And then when they're synapsing, there's a crossover that happens where these chromosomes are going to overlap, and then there's going to be an exchange of genetic material between the chromosomes so that we are not carbon copies of our own parents. And I'll show you a picture of that when we get to the next slide. That's what happens in prophase 1. So again, when we go through the rest of the stages, you'll find that everything is pretty much very basic, but all of the action that happens in the meiotic division occurs in your first prophase. Now, metaphase is where the chromosomes line up in the middle. So remember M, midline for metaphase. And then when that happens, you're going to, when you start pulling them apart, so apart would be your anaphase, where you're separating now these homologous chromosomes from each other. But again, you end up in your telophase, where the cell divides, making 2 daughter cells, but these daughter cells are haploids, meaning that now they have 23 chromosomes. But the problem is that each chromosome is still made out of 2 chromatids. So we have to go to your second meiotic division. Before we do so, we said we're going to explain better the process of synapsis and crossing over. So again, this is a pair of homologous chromosomes, which again is just a fancy term that means that both of these chromosomes, one from mom and one from dad, carry the genes for the same characteristics. So these would have maybe the genes for hair color on both of those spots, or the genes for maybe skin color, and so on. As long as they carry the same, they code for the same genes, they are termed homologous chromosomes. And when we start the whole process, your homologous chromosomes are going to come together and they are going to be paired. And we call that pairing synapsis. And then you'll see that there's a crossover where these chromosomes are going to kind of hold hands, so to speak. And then you'll see now that there's a exchange of genetic material. So you'll see now this X chromosome has part light blue and part dark blue. This part dark blue came from the other chromosome. So that again, this gives that genetic variation where when we end up making either a sperm or egg, it is not the exact copy of the parent. So when fertilization happens and babies are born, they're not carbon copies of their parents. Okay, so again, this all happens in the first meiotic division, to be specific, during prophase 1. Now moving on to second meiotic division, you have the 4 phases, and now you see here that they're just labeled to differentiate them between prophase 1 and prophase 2, and so on. At the end of both meiotic divisions, you'll end up making 4 cells. If this was happening in a testis and a sperm is being made, we end up making 4 sperms. Okay, but if this was happening in an ovary, we end up actually making 1 egg, just 1 egg. Now, what happens to the other 3 cells? Those degenerate. Okay, so again, in the process of spermatogenesis, which is the process of making sperms, the meiotic division will end up making 4 sperms. But in the process of oogenesis, which means to produce eggs, we'll end up making just 1 egg cell, and the other 3 cells are going to degenerate. This again shows you the crossing over that happens in meiosis. So here are the maternal chromatids, and these are the paternal chromosomes. And you can see here that they, the code, they're homologous chromosomes because they have the genes that code for the same thing. And then we have what happened is the synapsis and the crossover of genes. So now we have a good variation of genes, and this is the end result of the first meiotic division. We have haploid cells, so half of the number of chromosomes that we started off with, but again, your chromosome has 2 chromatids. So we have to go through the second meiotic division where now we make 4 cells. They're all haploid cells. But now each chromosome is made out of just 1 chromatid, and that is what you want to have. Again, in spermatogenesis, you'll end up making 4 sperms. In oogenesis, you'll end up making just 1 egg, and these 3 are going to degenerate. Now again, this is where I recommend where you kind of take a break, a mental break, maybe for a couple minutes, gather your thoughts on meiosis, and then come back and we'll start off with the organs of the male reproductive system. So what are the functions of the male reproductive organs? First off, to develop and to make sperms, and then to transport the sperms and the fluid known as semen to the outside of the body, and to make the male sex hormones. Okay, the primary sex organs or the gonads are the testis, and that's where the sperm cells or the spermatozoa and the androgens, which are the male sex hormones, are going to be produced. Any other organ besides the testis are known as the accessory sex organs, and some of them are internal, inside of the body, and others are external or outside of the body. So this is a figure that shows the different parts of the male reproductive system. And as we mentioned, that the primary organ or the gonad is the testis, and everything else is considered an accessory organ. So we'll go kind of run through the internal organs first and then the external organs. So the internal organs, we have the epididymis. Now the sperms are made in the testis, but they are immature. So they have to mature, they have to grow up inside of the epididymis, where they become the process known as spermiogenesis, where the sperms mature into a mature spermatozoa. And that occurs again inside of the epididymis. It is stored there until ejaculation will happen, and then it'll have to pass through the vas deferens or the ductus deferens. And you see here that it passes above or superior and then posterior to the urinary bladder. And these sperms are going to be dumped into an ejaculatory duct. As it goes through that duct, some fluids or some secretions are going to be added to those sperms. So for example, you have secretions coming from the seminal vesicles that also are emptied into the ejaculatory duct. The prostate that is surrounding the ejaculatory duct will also make secretions, and all of these secretions are going to be added here or pass through this tube known as the urethra. So the urethra is an organ that is a shared organ between the urinary and the reproductive system. There's a little tiny gland here known as the bulbourethral gland, and that secretes mucus. Now all of these secretions during ejaculation are going to will pass through the urethra until they're emitted out of the body through the penis. The external organs are the ones that we can see from the outside, so that includes the scrotum and the penis. Here again is another view of the different organs. This is looking at the organs from a posterior view. And the reason we know that this is a posterior view is because the seminal vesicles are found on the back of the urinary bladder. Okay, so again, you want to be familiar with the different parts and how a sperm being made in the testis. You want to be familiar with the different, the journey of it and until it gets out of the body. So we'll go through that journey, kind of fast one more time. So it'll start off in the testis, into the epididymis where it matures, and then it is stored there until ejaculation will happen, and then it'll pass through the ductus or the vas deferens into the ejaculatory duct. Now, when it goes into the ejaculatory duct, you have some secretions that'll be poured onto it. So you've got the secretions of the seminal vesicles. We have there's a pair, so right and left. And then there's one prostate gland. So this is a section in the prostate gland so that you could see the ducts inside of it. But the prostate gland is a single gland, only one gland in the body. And then there's the bulbourethral glands. Those are bilateral. And now all of that is going to pass through the urethra until it reaches the external urethral orifice. Now we'll talk about the descent of the testis, and this happens in utero. So as a fetus is developing, if the fetus is a male, the testis is actually, the cells originate from a mass of tissues that is found behind the peritoneum. It's found very close to the kidney. So these cells are retroperitoneal cells that are in close relationship to the kidney. And then 1 to 2 months before delivery, okay, so now again, this is all happening in the during fetal life. 1 to 2 months before delivery, testosterone is produced by the testis, and it triggers the descent of the testis into the scrotum. So you'll see here that the testis is made all the way up in the abdominal cavity, and then it has to be pulled down into the scrotum a couple of months before delivery. It is guided by this ligament known as the gubernaculum. This is a word you want to be familiar with, and the gubernaculum is going to pull the testis down, and as it's pulling the testis, it'll pull with it its blood supply. So there will be an artery and a vein, also its lymphatic supply or lymphatic drainage, and its nerve supply as well. And that's why you'll find that the artery, the blood vessels that supply the gonads come all the way up from very much related to the renal arteries and the renal veins. And that is, why this occurs, because of the embryonic origin of the testis. Again, the gubernaculum is going to keep on shrinking and pulling the testis down into the scrotum. Okay, that, all of these structures that are being pulled down, so the blood vessels, the lymphatics, the nerve supply, even the vas deferens, all of those together collectively are known as the spermatic cord. Okay, that route that it takes in order to reach the scrotum is known as the inguinal canal. Okay, and this is it's a weak area in the male abdominal wall, and that can lead to the high incidence of inguinal hernias in males. Now for the structure of the testis, it is surrounded by a fibrous capsule known as the tunica albuginea. Okay, and then inside of the testis, there are several walls, septa made out of thin connective tissue, and it divides the testis into very tiny lobules or tiny compartments, so about 250 compartments each. And then inside of these compartments, we have little tiny tubules that are very highly coiled, and those are known as the seminiferous tubules. These tubules are lined by the stratified epithelium, and they have the spermatogenic cells. So these are the cells that are going to eventually go through the steps of spermatogenesis in order to make a sperm. In between these seminiferous tubules, we have cells known as the interstitial cells or the cells of Leydig, and those cells produce and secrete the male sex hormones. So here you can see an image of the structure of the testis. So again, you have the capsule on the outside known as the tunica albuginea. And then on the inside, we have these different septa that divide the testis into compartments or lobules. Each compartment has these seminiferous tubules. If you take a section in a seminiferous tubule, so you take one of these and we made a cross section in it, you can find a basement membrane lined by all of these spermatogonia. Spermatogonia basically means that these cells are going to make sperms. Okay, so these cells are going to make, will go through the process of spermatogenesis in order to end up making the sperms. In between the seminiferous tubules, you'll have these cells known as the interstitial cells of Leydig, and those are responsible for the production of male sex hormones. Now, formation of sperm cells. Okay. So we already mentioned that the spermatogonia are the primitive cells that are going to start this whole process, but they need help, they need support, and that support comes from these large cells known as the sustentacular or Sertoli cells. So it helps that they all start with S. Okay, so the process of spermatogenesis starts off as the first cell to start off the process is known as a spermatogonia. So again, we're going to make sperms. And it needs support by these sustentacular or Sertoli cells. Okay, and they support the process through providing the nourishment needed for these spermatogenic cells to develop into sperms. So spermatogonia will develop into primary spermatocytes. Now these primary spermatocytes will further divide into secondary spermatocyte, and they do these primary spermatocytes will undergo the first meiotic division to produce a secondary spermatocyte. Now the secondary spermatocyte will undergo second meiotic division to make a spermatid. A spermatid is basically an immature sperm, okay, and I remember it like it's a kid sperm, so spermatid, and then it has to mature through a process that we termed spermiogenesis until it makes that mature spermatozoa. Okay? And so these definitions are things that, again, you want to be able to differentiate between those two terms. So spermatogenesis is the whole process where the cells went through the meiotic division and have matured into these mature sperms, while just the maturation process is known as spermiogenesis. Okay. Here you can see them kind of in an image where we start off with, actually, if you want to add here the cell, the spermatogonia, that would be great because the spermatogonia will undergo mitotic division to make tons of primary spermatocytes. Now the primary spermatocyte will undergo the first meiotic division, so primary, which means the number 1 cell, undergoes the number 1 meiotic division to become your secondary spermatocytes. And you see here that the secondary spermatocytes are made out of 23 chromosomes. Each of them has 2 chromatids. It undergoes its second meiotic division to make a spermatid. And you've now made 4 haploid cells. Each one of them has 1 chromatid only. And then the process of maturation of the spermatid into a mature sperm cell, and we named that process spermiogenesis. So again, you want to know the difference between these two processes. And here you can see what happens kind of like in that seminiferous tubule, where this right here is your basement membrane, and you can see the different steps of spermatogenesis as they are occurring. And as you go towards the center of the seminiferous tubules, you'll see that the maturation process is going further. So the very first steps, like the spermatogonium and the primary spermatocyte, those are going to be closer to the basement membrane. But the further away from the basement membrane you get, the further down the road you are in the process of spermatogenesis. And you'll find here that throughout this whole process, everything is being supported by your sustentacular cells. So let's talk about the structure of a sperm cell. So now that a sperm has been made and it is a mature sperm, what does it really look like? So is there a picture? No. Okay, so there's a head of the sperm that has a nucleus, and that again, the nucleus are haploid, so they're 23 chromosomes. And there's an acrosome. An acrosome is kind of like a little cap found on top of the nucleus. It has lots of enzymes, and those enzymes are going to be used in order to reach the egg during the fertilization process. There's a mid-piece that connects the head to the tail, and that mid-piece has lots of mitochondria. We'll use that energy, or the sperm will use the energy in order to swim. And the tail or the flagellum has lots of microtubules, and it'll use these microtubules for the swimming process as it tries to get to the egg. Now for the internal accessory organs in the male reproductive system, these are basically there to add some secretions to the sperms that the sperms are going to need on their journey, and also for transportation of semen to the outside of the body. So we'll start off with the epididymis, and we already named them, so I want to kind of go through the function, the structure and the function of each of them. So we'll start off with the epididymis. The plural is Epididymides, the singular is epididymis, so I usually use it in the singular form because it's a lot easier to pronounce. And it's basically a muscular tube that connects the testis to the vas deferens. In that tube, there's the maturation of the sperm cells, and that tube is lined with pseudostratified columnar epithelia. The cilia, though, there is non-motile, so it's not going to help in the, it's not going to push the sperms anywhere. The cilia are just there, but they are non-motile. The ductus deferens, you can see here a histological picture of it, where all of this is basically made out of smooth muscle fibers, and the lumen is very narrow. It is a muscular tube, is about 45 centimeters long. It is part of the spermatic cord, and it extends from the epididymis all the way to the ejaculatory duct. It is lined by pseudostratified columnar epithelium. Now for the first set of glands that are going to be reached, which are the seminal vesicles. Those, again, are bilateral, so there's one on each side. It is attached to the base of the ductus deferens. It is found posterior to the urinary bladder, and the secretion that it makes is an alkalinic fluid. And because the female reproductive system is a very acidic environment, so the seminal vesicles are going to make an alkalinic fluid in order to balance the pH. It also secretes fructose and prostaglandins, both of which help in the process. Sperms do not have the ability to produce energy out of glucose. They can only produce energy out of fructose. So they get that fructose from the seminal vesicles. Prostaglandins are important for the stimulation of, or for the contractions in the female reproductive system, and it helps in the fertilization process as well. Now, the fluids made by the seminal vesicles are going to be dumped into the ejaculatory duct, and they contribute most of the volume of semen. So when we talk about semen and what is it made out of, we'll find out that the majority or most of the fluid, that white fluid, is mainly made out of the fluids coming out of the seminal vesicles, volume-wise. Color-wise, though, that's a different story, in that which takes us to the prostate gland. The prostate gland is found, it surrounds the upper part of the urethra. In the middle, you'll find the urethra and the ejaculatory duct. It definitely contributes to some extent to the volume of semen. It secretes a thin, milky alkalinic fluid, so that milky color of semen is really because of the secretions of the prostate gland. Again, the alkalinity is there to balance the acidity of the vagina, and the secretions that are made help in enhancing sperm motility. So as the sperms are being ready to be ejaculated, they have to be activated. And a lot, the prostate secretions help in the process of activation of sperms. Now, since we're talking about the prostate, we want to talk a little bit. Sometimes it does not even require treatment, but in other cases, it could be metastatic and fatal. It is diagnosed by different things. So we start off by a rectal exam. So if you look back at the image of the male reproductive system, you'll find that this is the prostate, and right here you'll find this is where the rectum is. So doing a PR or a rectal exam, if there's an enlargement of the prostate gland, sometimes that could be felt that way. And the patient sometimes complains of frequency in urination, meaning that they have to go to the bathroom very frequently, and a slow process of urination where it's hard for them to urinate. Like they feel the urge to urinate, but they have to, push very hard in order to overcome the enlarged prostate. So if you have the anatomy in your head, if the prostate is enlarged, it might block the passage of urine through the urethra, so that it leads to this frequency in urination and being difficult to urinate. There's a blood test that can be done that is to detect this PSA, which stands for the prostate-specific antigen. It helps in diagnosis, but you want to know that PSA is not specific for prostate cancer, because it can increase in inflammation of the prostate or prostatitis. It can even increase in what is known as BPH, which is benign prostatic hyperplasia, which is basically a normal process of aging in males where the prostate tends to enlarge as they age. So you want to kind of take take a look at the PSA levels, but you have to take the whole clinical picture into consideration. If, PSA levels are high and there is a good reason to do so, you would go ahead and take a biopsy, okay, and look at that biopsy and make sure that you kind of exclude the presence of prostate cancer. Some of the treatment options would be surgical removal, radiation, and hormonal treatment. Moving on to the bulbourethral glands. These are very small glands. They're called the Cooper's glands. They're found inferior to the prostate, and they produce a mucus-like fluid. And those they're stimulated by sexual stimulation, and they help in the lubrication process. They lubricate the end of the penis. So if you look at all of the fluids that make semen, okay, semen is the fluid that is going to be projected or ejected out of the body during ejaculation. It is made out of sperms and the different secretions. So we have your sperm cells, they're the secretions of the seminal vesicles, excuse me, the prostate and the bulbourethral glands. It also includes prostaglandins, and it is slightly alkalinic. So remember that the seminal vesicles and prostate glands, those both are alkalinic fluids. The volume per ejaculation is about 2 to 5 milliliters of semen. If you're not sure how much that is, 5 milliliters is a tablespoon. And per milliliter of semen is about 120 million sperm cells. Okay, and sperms, as I said, when they're made, without these secretions, they won't be able to swim in the absence of the secretions of all of these accessory organs. Now, male infertility is the inability of the sperm cells to fertilize an oocyte. Some of the causes could be failure of the testis to descend in the scrotum that, during fetal development. Could be due to inflammation and destruction of the testis from certain diseases and infections, like mumps, for example, which is a viral infection of childhood that could lead to the inflammation and destruction of testicular tissue. Poor quality sperm, so the sperm might be made, but it has an abnormal head or abnormal parts. Or the sperm is normal, but there's a low sperm count. So anything less than 20 million per milliliter is considered low sperm count. CASA, or computer-aided sperm analysis, this is a technique to evaluate male infertility. It basically looks at sperm count, sperm motility, size, and shape of the different parts of the sperm. Now for the male external reproductive organs, namely the scrotum and the penis. And we'll talk about each one of them. So the scrotum is that pouch of skin and subcutaneous tissue that is found behind the penis. The subcutaneous tissue does not have fat in this area. There is a smooth muscle though, and that smooth muscle is known as the dartus muscle, and it contracts and relaxes in response to changes in temperature. So in order for the testis to survive, it has to be at a colder temperature, and that's why it is found outside of the pelvic cavity. It has to be about 5 degrees below the core temperature. Core temperature is the temperature inside of the body. So in very cold temperatures though, in order to protect the testis, the dartus muscle contracts and brings the testis to the inside of the body where it could remain warm. And then while in warm weather, the dartus muscle relaxes and the the testis will go back into the scrotum. The testis is divided by a medial septum into 2 chambers, so a right and a left side, and each chamber has its own serous membrane, and each chamber houses a testis and an epididymis. Now for the penis, that's where the urethra is found, and again, it is an organ that is shared by the urinary and the reproductive system. The body has the erectile tissue, so we have 2 corpora cavernosa and 1 corpus spongiosum. The corpus spongiosum is the one that surrounds the urethra, and we'll take a look at it at the next picture. Then there's a glans penis, which is the distal enlargement at the tip of the penis of the corpus spongiosum. And there's the foreskin, or the prepuce, which it covers the glans penis and is removed during circumcision in some individuals. So here, if you take a section of the penis, this is where the urethra is found in the middle, and it is surrounded by the corpus spongiosum. So in order to remember it, you want to remember that the urethra wants to be protected, so there's a sponge surrounding it. So this corpus again means body, spongiosum means the spongy body, and that is the body that surrounds the urethra and protects it. These two are known as the corpus cavernosa. Okay, cavern comes from the word that is very highly vascular. It has a high blood supply. So you'll find these 2 arteries, and when an erection happens, these arteries dilate, and that's why lots of blood reaches the area. And because of there's lots of blood coming in, it compresses the veins. The veins are compressed while the arteries are dilated, meaning that blood can get into the area, but it cannot flow out. And that leads to the accumulation of blood in the area, leading to an erection. Now for erection, orgasm, and ejaculation. The very first process, okay, it starts off as a parasympathetic stimulation. And parasympathetic is going to release the local nitric oxide, and nitric oxide dilates the arteries in the penis. With the pressure of the arterial blood, that's going to compress the veins. So again, you have the arteries dilated, allowing for blood to come in, while the veins are compressed, preventing the blood drainage, and that leads to accumulation of blood in the tissue, eventually leading to an erection. While the ejaculation process and orgasm, that is due to sympathetic nervous system. Okay, so you'll find that there is tachycardia, tachypnea, which means increase in the rate of breathing, and so on. That is due to the sympathetic stimulation. So the whole process starts as parasympathetic stimulation, while it ends with a sympathetic stimulation. Now, emission is the movement of semen into the urethra. Okay, and that again happens by the contraction of the smooth muscles for the sperms to be transported through that process that we talked about prior. And ejaculation occurs due to, or is defined as the movement of semen out of the urethra. So what is the hormonal control of the male reproductive function? Well, everything starts off again with the hypothalamus, and there's this whole loop system between the hypothalamus, anterior pituitary, and the testis. The hormones that come out, they are going to start the development and maintenance of male secondary sexual characteristics. So the development means, and that's going to happen during puberty, where all of the changes occur, while the maintenance, meaning, over the rest of their lives, where these sexual characteristics are maintained. Now, for the different hormones that come out, the hormone that comes out of the hypothalamus is the gonadotropin releasing hormone that will stimulate the anterior pituitary to make LH, luteinizing hormone, and FSH. Okay, luteinizing hormone is sometimes in males referred to as interstitial cell stimulating hormone, but it's the exact same hormone. It just has these two different names. The LH in males is going to stimulate the development of the interstitial cells of Leydig, and we kind of talked about those already. And those cells are going to secrete testosterone, while the FSH that comes out of the anterior pituitary is going to stimulate the sustentacular cells in order to help in the process of spermatogenesis. By negative feedback, you'll see that the production of androgens are going to prevent the oversecretion of gonadotropin-releasing hormone from the hypothalamus and luteinizing hormone from the anterior pituitary, while inhibin, which is a hormone produced by the sustentacular cells, will prevent the oversecretion of FSH. So you can go to this diagram and take a look at that whole feedback loop. So your hypothalamus secretes gonadotropin-releasing hormone that stimulates the anterior pituitary to make both FSH and LH. Now, FSH through the bloodstream will reach the testis, and it'll stimulate the whole process of spermatogenesis. So FSH, which stands for follicle stimulating hormone, which in females will stimulate the process of making the mature egg. It does the same in males, so it'll also stimulate the process of making a mature sperm. So FSH again reaches the testis and stimulates the process of spermatogenesis, while luteinizing hormone also reaches the testis and it stimulates the making of testosterone. Now, testosterone and all of the other androgens are going to be made by the testis and circulate through the system. By negative feedback, they are going to inhibit both the hypothalamus and the anterior pituitary. While inhibin, that's a hormone made by the sustentacular cells, are going to, that inhibin will circulate to the anterior pituitary and prevent the oversecretion of FSH. Now, for the function of androgens, and notice that androgens is plural. There are a lot of different androgens or lots of different male sex hormones. The most important is testosterone, and those are going to produce these following effects. So they increase muscular growth, increase hair growth on the face, chest, axillary, and pubic areas. It will start the thickening and strengthening of bones and thickening of the skin. It leads to the enlargement of the accessory reproductive organs. It also leads to the enlargement of the larynx and thickening of the vocal cords, leading to that change in voice during puberty. So your male sex hormones, those are known as androgens. As I mentioned, the most important of them is testosterone. They're made by the interstitial cells. The secretion begins during fetal development. So in utero, we mentioned that during the last 2 months of pregnancy, the baby testis is going to produce some testosterone, and that helps with the descent of the testis. And then secretion is going to stop throughout, after delivery and then throughout childhood and so on. And things are not going to kick in again until puberty, and then it continues throughout life. So the action of testosterone, we already kind of talked about most of them. So prior to birth, it would be the development of the male reproductive organs and the descent of testis. During puberty, we have the primary sex characteristics, so the enlargement of the testis, and it helps in the development of the secondary sexual characteristics, and we already, mentioned those before. And in males, testosterone helps increase the rate of red blood cell production and cellular metabolism, and that's why we've mentioned that, generally speaking, that the RBC count in males is higher than it is in females, and that metabolism is higher in males as well due to the higher levels of testosterone. So if you're listening to this lecture, this is where I would definitely recommend where you would stop, take a break, come back later to move on to the female system. Okay, so we're back and we'll start talking about the organs of the female reproductive system. The function would be to produce the female sex cells, the oocytes or your egg cells, to transport the oocyte to the site of fertilization. And then there's a good environment for the development of offspring, namely the uterus, and then the transportation of the offspring to the outside of the body during delivery. It also helps in the production of female sex hormones. The female sex organ or the primary organ would be the ovaries, and then everything else in the system are considered accessory organs. And again, there are some of them are internal, so those are inside of the body, while others are external. And we'll take a kind of a overview of the different organs. So we'll start all the way here with the ovaries, and then there's the oviduct, sometimes called the fallopian tube, and that will take us to the uterus. And then the exit of the uterus is known as the cervix, and that's the part of the uterus that is found at the upper portion of the vagina. And then there's the vagina and the external vaginal orifice. For the external parts, there is the labia majora and the labia minora, and the clitoris and the vestibule. You really cannot see those from this view, but we'll take a look at it when we get to other slides. The ovaries are pretty much need some ligaments in order to attach them, because they are found on the sides of the pelvic cavity, and they need these ligaments to hold them in position. So the biggest of these ligaments are the broad ligament, the suspensory ligament, and the ovarian ligament. Okay, the broad ligament is the biggest of all three. It holds the ovary in place, and it also is attached to the uterine tubes and the uterus. There's the suspensory ligament that holds the ovary or kind of suspends it from its upper half. And then there's an ovarian ligament that attaches the lower end of the ovary to the uterus. So we'll take a look at them right here. So this right here is the broad ligament, and that is the biggest of these ligaments. You can also see the suspensory ligament of the ovary, and that literally suspends it from the pelvic bone. And there's also the ovarian ligament. And you see that the pictures are a little bit different, so we kind of pushed the fallopian tube away so that we could see the ovarian ligament connecting the ovary to the uterus. The ovaries have to descend as well, just like the testis does. The ovary is also made from tissue that is found very near to the kidney, behind the peritoneum. They need to descend to their pelvic, to where they are in the pelvic cavity, but they do not descend out of the pelvic cavity. So unlike in the male system where they descend down to the scrotum, the ovaries go all the way to the pelvic brim. Now for the structure of the ovary, there's a cortex on the outside, and that's where the cells are multiplying and maturing into these different Graafian follicles, while there's the medulla on the inside, and the medulla has the blood vessels and the lymphatics and the nerve supply. We'll talk about these little primordial follicles. Now, oogenesis is, although, it does follow the primary meiotic division and the secondary meiotic division, kind of like in the process of spermatogenesis, but time-wise, it's a little bit different. Okay, so the first meiotic division actually starts in utero. So during prenatal development of a female germ cells, the oogonia, okay, again, these are going to make eggs, we'll start the first meiotic division, and they are going to develop into these primary oocytes, and they remain stuck in the middle of meiosis 1. Okay, now these baby girls are born with these primary oocytes that are right in the middle of meiosis 1, and they are going to remain in that, as such until puberty starts. So these primary oocytes are surrounded by lots and lots of epithelial cells known as follicular cells, and we're going to call that primary oocyte surrounded by these follicular cells, we'll call them a primordial follicle. Now that primordial follicle has to develop into your mature Graafian follicle, but that again is not going to happen until puberty occurs. So once all of the primordial follicles are made and the baby is born, there is no way to add new primordial follicles. And that is why once those primordial follicles are all gone, that's why menopause occurs. So unlike in males where spermatogonia can always or have that ability to replenish themselves, oogonia do not. Okay, so oogonia, because they've already started that first meiotic division in utero and they are stuck in that first meiotic division, after birth, there is no way to replenish them. Okay, so the number of oocytes are going to decline even before puberty hits. So as about 90% of the primordial follicle at birth are lost to apoptosis between birth and between early adulthood. So the remaining 10% is what lasts throughout the what is kind of known as the reproductive years. And then when all of those are gone, menopause occurs. Now, oogenesis, that is the scientific term for the process of making eggs. Remember, it says we started off in utero, but we're going to continue what we started. Beginning at puberty, some of these primary oocytes are going to continue through the first meiotic division and will make a secondary oocyte and a first polar body. So remember, we said that when we're making eggs, we end up making just one egg, and the remaining 3 cells are going to disintegrate or die off. These 3 dying off cells are known as polar bodies. And the function of these polar bodies is basically just to get rid of all of that extra genetic material that we are no longer in need of. Okay, so at the end of the meiosis 1 will have a secondary oocyte and that first polar body, and that will just die. Now the secondary oocyte is going to remain there, and secondary or meiosis 2 only occurs if the egg is going to be fertilized. So if no fertilization occurs, second meiotic division does not occur either. Okay, so secondary oocyte will undergo meiosis 2 only if fertilization happens. And that is going to lead to, that cell being fertilized by the sperm, leading to a zygote or a fertilized egg, and then we'll make a second polar body that again is going to disintegrate. These polar bodies are only there in order to take away that extra genetic material that we don't need. Here you can see in picture what happens. So your primary oocyte starts off the first meiotic division, and it'll continue to make a secondary oocyte. You'll see, and a first polar body. The first polar body again is there just to get rid of that extra genetic material. Your secondary oocyte is not going to go through second meiotic division unless fertilization happens, second meiotic division occurs, fertilization happens, and we end up having a zygote, while the second polar bodies are going to disintegrate. Now for the maturation of the follicle, okay, at puberty, the hypothalamus makes gonadotropin-releasing hormone, and that is stimulate the anterior pituitary to make FSH and LH. FSH, named follicle stimulating hormone, is now going to stimulate these primordial follicles in order to mature into that mature egg. Okay, it'll lead to the enlargement of the primary oocyte. The cells surrounding it, the follicular cells, will start to proliferate into several layers, and those layers are known as the granulosa cells. We also develop a zona pellucida, which is a pale zone made out of glycoprotein, and it forms between the oocyte and the granulosa cells. So we'll take a look at what this looks like. Do we not have a picture? Okay, I'll go back to those in a minute. So this right here is a secondary oocyte. This is what a mature Graafian follicle is. Okay, there's a secondary oocyte here. We have this very thick layer of theca externa layer. And then we have all of these follicular cells surrounding it. And there's a little part here of an antrum that is full of fluid. This is what is going to be ovulated. Just this portion right here. When ovulation occurs, what will be ovulated is the secondary oocyte with that white zona pellucida surrounding it and some of these granulosa cells. The remaining of this Graafian follicle is going to change into another structure known as the corpus luteum. The corpus luteum or the corpus luteum will secrete progesterone. Okay, so this mature follicle secretes estrogen. After ovulation, which again the ovulation would be the release of the secondary oocyte, the zona pellucida, and some of the follicular cells surrounding it. That'll be ovulated outside of the ovary. And the remnants of this will change into a structure known as the corpus luteum and will secrete progesterone. So these are the different stages of follicle maturation. We end up with a primordial follicle that changes into a primary follicle, and then we have your mature antral follicle, and we already saw the different parts of the mature antral follicle. And this is when ovulation happens. So again, the ovulation would be the secondary oocyte, the little zona pellucida, and the whole bunch of follicular cells surrounding it. The remnants are known as the corpus luteum. If fertilization does not occur, this corpus luteum is going to degenerate into fibrous tissue and is now a white body, so we call it a corpus albicans, and it'll just die off, and it has no function. Okay, but if fertilization does occur, this corpus luteum remains, keeps on secreting progesterone in order to maintain the pregnancy for about 3 months until the placenta is fully formed, and then the placenta can take over and make enough progesterone to sustain the pregnancy. When a follicle develops, we have these different layers. So there's the inner vascular layer known as the theca interna, makes steroids, and the outer vascular layer or the theca externa, and that's made out of connective tissue and it's protective. Lots of follicular cells, and then there's that fluid space, that fluid-filled space. Okay, in order for the whole maturation process to happen, it takes about 150 days. So the follicle that is ovulated today did not really start a month ago. It started about 150 days ago. But because of this being a cycle, every month a follicle is released. Okay. The the mature follicle is known as the Graafian follicle. Graaf is the scientist that discovered it and was named after the scientist. So Whether you call it the Graafian follicle or the mature follicle, they're both the same thing. Okay, so because I did not mention what the corona radiata was, the corona radiata would be these, it's kind of like a crown that is surrounding the zona pellucida. It is made out of the follicular cells, and again, it's part of what is ovulated. So again, what is ovulated is your secondary oocyte, the zona pellucida, and the follicular cells, and we call these follicular cells, we call them the corona radiata. Okay, the follicle maturation process, usually only one of them, lots of different follicles are stimulated, but only one of them eventually will reach the full maturation. The continuous, again, like I said, because it's a very continuous process that keeps on occurring in a cycle, so that every month there is a mature follicle, or there's one follicle at least that reaches maturation. Now for the process of ovulation, near the end of a follicle's maturation, we mentioned that already, the primary oocyte will complete meiosis 1 and become a secondary oocyte. Like I said, at ovulation, the secondary oocyte is what is released, surrounded by the zona pellucida and the corona radiata. But secondary oocyte is not going to go, or will not undergo, the second meiotic division unless fertilization happens. Now for the different parts of the internal reproductive system in females, we'll talk about them, structure and function of them as we go. Here, this is you can see a section where you can find the ovary, and then there are these fimbria, which are finger-like projections surrounding the ovary, and they help in picking up that egg as it is ovulated, so that the egg doesn't fall into the pelvic cavity. It is rather picked up by the fimbria and remains in the infundibulum. The infundibulum is that funnel-shaped area, and this is where fertilization occurs. So the sperm would have to, swim throughout all this journey right here in order for fertilization to happen in the infundibulum of the fallopian tube. And then there's the fallopian tube, or the oviduct, and then the uterus. Implantation occurs in the uterus. And you can also see the cervix, which is the exit of the uterus, and it invaginates into the upper part of the vagina. Now we'll talk about the uterine tube, or the fallopian tube, or the oviducts. All of these are just three different terms for the same thing. It is that tubular organ that transports the ovulated egg from the infundibulum all the way into the uterus. It is lined by little cilia. Okay, the mucosa is lined by cilia, and that helps the transportation of egg down the uterine tube. Okay, like as we mentioned, that the uterine tube has fimbria, which are the finger-like projections that pick up the egg after fertilization, and then the fertilization occurs in the infundibulum area. The uterus is a very muscular organ, and it will receive the embryo and sustain it and protect it during development. It is attached to the pelvic wall by the broad ligament and a round ligament. The regions of the uterus, there's the body that makes the upper part, or the dome, also known as the fundus. And there's a cervix, or a neck. That would be the lower half. And that, like I said, partially extends into the upper part of the vagina. And this is where, when a pap smear is done, that is a smear or taking off cells from the cervix in order to check for cervical cancer. The different layers of the uterus, there is the mucosal layer that's known as the endometrium, and that is the layer that is shed every month during menstruation. There's also the myometrium, which is the muscular layer, and then the perimetrium, and that is the serosal covering. So I'm going to go back to this image to show you the different layers again. So the endometrium is the inner layer, and that is, again, that's the layer that is shed during menstruation. The myometrium, that's the strong, smooth muscle that contracts during delivery. And then the outer layers, or the perimetrium, that's the serosal layer, and that you can see the blood supply through that layer as well. Now, the vagina is a fibromuscular tube, and it runs between the uterus and the outside of the body. It'll convey uterine secretions out of the body. It receives the penis during intercourse, and it's a passageway for offspring during birth. At the very top, it surrounds the end of the cervix, and the parts, the recess between the vaginal wall and the cervix is known as a fornix. I'll show you that fornix in a better picture right here. Okay, so that little angle between the cervix and between the vagina, that is known, those are the fornices that we were talking about. The vaginal orifice is partially enclosed by a hymen, which is a thin layer of connective tissue, and stratified squamous epithelium, kind of like skin. And the wall of vagina has 3 different layers. There's the inner mucosal layer made out of stratified squamous epithelium, and then there's a middle muscular layer and an outer fibrous layer. For the external reproductive organs, we already mentioned the labia majora, there's the labia minora, and then the clitoris, and we talked about the vestibular area. And the vestibular area would be the area between the 2 labia minora. So we'll take a look at the structure and function of each of them. Labia majora are made out of skin with the, there we go. So the labia majora is the skin that's lies over adipose tissue, and they merge in the front to make this mons pubis area. The mons pubis is has the pubic hair as well. And then the labia minora, those are very full of nervous tissue. They're full of blood supply as well. And then at the very front, they combine to make the clitoris. The clitoris is a small projection, and it is found at the anterior end of the vulva. It has the erectile tissue that is found in the penis, so it kind of corresponds to the male penis. And during sexual stimulation, it is erected because of that erectile tissue. The vestibule, that would be the area between the 2 labia, and it encloses, you'll find the opening of the vagina and the opening of the urethra. You'll also find the vestibular glands that will make mucus, and these vestibular glands correspond to the bulbourethral glands in males. Now for erection, lubrication, and orgasm. In erection, like I said, the erectile tissue found in the clitoris do respond to sexual stimulation. The nitric oxide that is released will lead to the erection of that tissue and leading to the expansion of the vagina. Then there's lubrication due to the vestibular glands that release mucus. And then there's orgasm, where also due to the sympathetic stimulation and leads to the peristaltic contractions of the perineal muscles, the uterus, and the uterine tract tubes. And those are going to be helping in the transport of the sperm towards the egg. So hormonal control of the female reproductive functions. The hormones that are made by the hypothalamus, and then the anterior pituitary, and then the ovaries. So that is kind of like the 3 different parts. They help in the development and maintenance of the female sexual characteristics, maturation of the female sex cells, and the changes that occur during the menstrual cycle. So we'll talk about gonadotropin releasing hormone. The exact same thing happens in females, where it will circulate to the anterior pituitary, stimulating it to release FSH and LH. FSH, from the name, follicle stimulating hormone, is going to stimulate the maturation of the follicle, and that leads to the production of estrogen, while luteinizing hormone helps in the process of ovulation. And after ovulation occurs, the corpus luteum will start making progesterone. Now, estrogen and progesterone both are made by cells of the ovaries. They are also made to some extent by the adrenal cortex, and they're made by the placenta during pregnancy. Okay. Estrogens, the function of estrogen is that they stimulate the enlargement of the accessory organs. They stimulate the thickening of the endometrium, and they help in the development of the female sexual characteristics, namely the breast and mammary gland duct development, the increase of fat tissue in the breast, thighs, and the buttocks area, and increased vascularization of skin. While progesterone will stimulate different changes in the uterus, it leads to the increase of blood vessels and increase of the glands in the endometrium. It also affects the mammary glands, and it regulates the secretions of gonadotropins. So we'll see here how that loop works. Again, the hypothalamus making the gonadotropin releasing hormone to stimulate the anterior pituitary to make both FSH and LH. Now, FSH is going to stimulate the maturation process. Now that maturing egg will start making estrogen, while LH is going to stimulate the process of ovulation. When ovulation happens, now that corpus luteum is going to start making progesterone. Okay, now for the estrogens, through the bloodstream, will lead to the different changes that we just talked about, while it will help in the inhibition of the anterior pituitary and the hypothalamus, and that will lead to low levels of gonadotropin hormone. When levels of the hormones go down, that will lead to menstruation, and then this whole cycle will just keep on repeating every month. So the menstrual cycle is characterized by the regular recurring changes that occur in the endometrium, and at the end, if no pregnancy occurs, then will lead to menstruation. Menstruation usually, menarche, which is the first reproductive cycle, usually starts at the age of 13 and continues maybe into the late 40s, early 50s. It's very different from one person to another. Each month, there is one mature antral follicle that is ovulated. The remnants of that follicle, again, changes into this yellow body, the corpus luteum, that makes progesterone. In the absence of fertilization, that corpus luteum is going to degenerate and become fibrotic, leading to a white body known as the corpus albicans. So this is the whole list of changes that occur. I'm going to let you guys read this because we already talked about them, but they're found kind of like in this table. So you can go ahead and pause, go through it, and then move on to the next slide. These are the major events that happen in the menstrual cycle. I am going to go over them in a different slide, okay, as opposed to just reading it off of here. Okay. So these are the events that happen during the menstrual cycle. On average, the cycle is about 28 days long. Again, very different from one person to another, but we'll do the average, which is 28. And it is divided in the middle by this event known as ovulation. Okay, so there is a pre-ovulatory phase and a post-ovulatory phase. In the pre-ovulatory phase, excuse me, the anterior pituitary is mainly making FSH. So before ovulation, most of the changes that occur are under the effect of FSH. You'll see that in the ovaries, FSH is going to stimulate the maturation of the follicle. As this follicle is growing, it is also releasing estrogen. Okay, so we call this phase in the ovary, we call it the follicular phase because the follicles are being made and estrogen is being produced. Now that estrogen, you see these estrogen levels going up, will reach the endometrium, okay, and lead to certain changes in the endometrium. It'll lead to proliferation of the cells of the endometrium, which is basically mitotic division of the endometrium. And because it leads to the proliferation of the endometrium in the uterus, we call this the proliferative phase. Okay, so again, before ovulation, in the ovary, that phase is known as the follicular phase. Under the control of FSH from the anterior pituitary to mature that Graafian follicle, the follicle starts making estrogen. Estrogen levels rise. That estrogen reaches the endometrium, leading to the proliferation of the endometrium, and we name that the proliferative phase. Now that ovulation occurs, and ovulation happens because of that surge in LH. So that sudden increase in LH leads to ovulation. Now ovulation happens. This remnant changes into corpus luteum. Again, this phase is under the control of LH from the anterior pituitary. And because the corpus luteum is making progesterone inside of the ovary, we'll call it the luteal phase. That progesterone levels reach the endometrium, and it leads to the increase in the secretions of the endometrium. So these white spiral things that you're seeing, those are glands. It increases the glands of the endometrium, and it increases the vascularization, the blood supply of the endometrium. And that's why we call it the secretory phase. So your follicular phase in the ovaries correspond to the proliferative phase in the endometrium, while the luteal phase in the ovary corresponds to the secretory phase in the endometrium. Then when the corpus luteum degenerates and changes into the corpus albicans, there are no more estrogen or progesterone are being made, and due to the drop of these hormones, that endometrium is going to die off and eventually leading to menstruation. So you'll see here the menstruation that occurs as well, which is basically the endometrium dying and being sloughed off with some blood out of the body. Something to also keep in mind that the first day of the cycle, okay, is the first day of bleeding. So your part of this, the first half of the cycle is actually made out of the menstrual phase plus the proliferative phase. Okay, so if you're talking in the terms of what is going on in the endometrium, before ovulation, we have menstrual phase followed by proliferation or the proliferative phase. After ovulation, you have the secretory phase. And then at day 28, that's the end of the cycle, and menstruation starts again, and we start a new cycle. So that's day 1 again will be all the way over here. So we'll talk a little bit about female infertility, and that is the inability to conceive a child after a year of trying. Male and females contribute equally to the problem. 25% of the cases are due to more than just one cause. And some of the causes of female infertility would be anovulation, which means that there is no ovulation, which could be due to the hyposecretion of FSH and LH. Endometriosis, which is the endometrial growth of, or abnormal growth of endometrial tissue in areas besides the endometrium. So there could be some endometrial tissue in the abdominal cavity, for example, or in the uterine tubes, and that could lead to fibrosis and infertility. Repeated infections, such as gonorrhea, for example, can also lead to scarring of the uterine tube, leading to infertility. Now, menopause, that is the stopping of the menstrual cycle. It usually occurs in the late 40s or early 50s, where there is no more production of estrogen and progesterone because there are no more follicles. Some of the female secondary sexual characteristics start to disappear. Half of women during menopause experience what is known as hot flashes. Some experience other problems like migraines, backaches, and fatigue. Now, a little bit about the mammary glands, and those are part of the accessory organs in the female reproductive system, although the glands are found in males as well, but because of the lack of the estrogen and progesterone, they are not enlarged, but they do enlarge in females due to the presence of these female sex hormones. Okay, there is the nipple where we'll find all of these alveolar ducts open up and surrounded by a part of the skin that is a little bit darker in color, and that is known as the areola. Like I said, the mammary glands, the actual glandular tissue, is present in both male and female. At puberty, the ovarian hormones, so estrogen and progesterone, stimulate the growth or the development of the mammary glands in females. The structure of the gland. Each gland is made of about 15 to 20 lobules, and each lobule contains what are known as alveolar glands. So if you don't remember what those are, you can go back to chapter, I don't know whatever we were talking about, different kinds of epithelial and connective tissue and so on. You could go back and review what an alveolar gland was. Each gland has its own duct, and that duct is known as the lactiferous duct. There's also different strands of connective tissue, and those strands of connective tissue suspend the mammary tissue to the skin. You cannot see it in this image, unfortunately, but they're known as the suspensory ligaments. Now for a little bit about breast cancer. Some warning signs, things to kind of look at, or red flags. Thickening of the tissue, areas of thickened tissue, a dimple that was not there on the breast tissue, change in contour, flattened nipples, or a nipple discharge. Okay, biopsy can always confirm the presence or absence of cancer. Surgical treatment includes a lumpectomy, which is just the removal of the lump. A simple mastectomy would be removal of the breast tissue. A radical mastectomy would be removal of the breast tissue with the lymphatic tissue. We can also follow up with radiation and chemotherapy. Some of the things to keep in mind for early diagnosis would be self-breast examination and annual mammograms. Sometimes genetic testing can occur in order to detect the presence of different of breast cancer genes, especially if there is a strong family history. And about 5 to 10% of all breast cancers arrive from inherited tendencies. So birth control is the voluntary regulation of the number of offspring produced or the time when they are conceived. This controls, birth control is going to need some kind of contraception, and contraception would be either to prevent ovulation from occurring, or to prevent fertilization from happening, or to prevent implantation. Several different kinds, there are different options for contraception, so we'll take a look at some of these and kind of go over them and the different degrees of effectiveness. So there's coitus interruptus, which is the practice of withdrawing the penis from the vagina before ejaculation. Okay, so the thought is that, withdrawing the penis before ejaculation occurs, so the sperms are not going to reach the eggs and fertilize them. Problem is that it is usually it's not an effective way. Okay, sometimes, couples sometimes have a problem doing this. Another thing is that sometimes sperms actually leave the urethra before ejaculation. So there is some fluid that comes out of the penis before the actual ejaculation occurs. And that liquid, that fluid that comes out, some of it does have sperms already. So even if, they are successful in doing this, it might lead to still some sperms reaching there. So it's not a very effective way of contraception. There's a rhythm period. Rhythm period means that there is no sexual intercourse 2 days before and 1 day after ovulation. Again, it's not very effective. It really depends on a couple of things. For example, that the cycle has to be very regular. Okay, the couple has to be very careful in writing down when menstruation started and to journal everything to make sure that there is no intercourse that occurs during the ovulatory or during the ovulation. Other things that occur like mechanical barriers and chemical barriers. So in mechanical barriers, you're basically putting something to prevent the sperm from entering the female tract. For example, condoms. There's a chemical barrier as well, which is, for example, spermicides. Those spermicides, literally from the name, they are going to kill sperms. So they're in the form of creams, foams, or jellies. And these are most effective when they are used together with a mechanical barrier. There's also combined hormone contraception, so to give estrogen and progesterone to prevent pregnancy. So when you give them together, that is going to disrupt the whole negative feedback that we were talking about. So there's a change in the FSH and LH secretions, and those are going to prevent the follicle maturation and ovulation. Giving estrogen and progesterone also thickens the cervical mucus, so that kind of forms a plug-like mucus to prevent the sperm from reaching the egg. Different ways of delivering the hormones. So the most common way is through a pill, but there are other ways like injection or an intradermal patch. Other hormone contraceptions, like I mentioned, so there's an intramuscular injection, of medroxyprogesterone, which is just one of the progesterones, and that could, it's taken every 3 months. That'll prevent the maturation and the release of the secondary oocyte. An IUD is a mechanical barrier, which is a solid subject that is implanted into the uterine cavity, and there are different kinds of them. Some of them can remain in there for 5 years. Others can remain for 10 years. It all depends on what, on what the needs of the couple is. There's sterilization methods as well. Sterilization is different from contraception. So contraception is a temporary loss of the ability to conceive, while sterilization is a permanent loss of the ability. There is a vasectomy in males where there is the vas deferens is cut and tied. So basically the sperms are still made, in the testis, because the testis is fine. So the sperms are still made. They can still be stored here in the epididymis, but there's no way for these sperms to reach the outside of the body. In tubal ligation, in females where the fallopian tube, again, is cut and tied. So again, the oogenesis process, the process of making eggs, that has not been interrupted. The only problem is that here you've prevented the sperm from reaching the mature egg. Now, a little bit on STIs, or sexually transmitted infections. We tend to call them STIs now instead of STDs, because they could be, they are infections and they are contagious. So some of the symptoms of STI, some of the common symptoms would be burning sensation during urination, pain in the lower abdomen or pelvic pain, fever and swollen glands, so the lymph nodes. There could be discharge from the vagina or the penis. There could be itching, pain, and inflammation in the genital or in the perianal area, sores, blisters, bumps, or rashes, and itchy and runny eyes. So these are just general symptoms of STIs. There's a pelvic inflammatory disease, which is a complication that happens because of gonorrhea or chlamydia, where bacteria enters the vagina and it spreads to the reproductive organs and can lead to scarring tissue and fibrosis, leading to infertility. These are some of examples of STIs. So there's an HIV infection that due to the human immunodeficiency virus. That one of the complications of an HIV infection would be AIDS syndrome. Chlamydial infection, that is a bacterial infection that can be treated by antibiotics. Some of the complications are the PIDs that we talked about that can lead to infertility. Another complication is arthritis. So some of these patients might develop arthritis as a complication due to the bacterial infection. And because of the scarring tissue, there could also be an increase in incidence of ectopic pregnancy. Ectopic pregnancy means that pregnancy occurred outside of the uterus. Genital herpes, that is an infection by the herpes simplex 2 virus that leads to genital sores and fever. The effect on the fetus can lead to brain damage. It is treated by an antiviral drug, and there's an increased incidence of cervical cancer. Genital warts is by a virus known as the HPV, or human papillomavirus, that also increases the risk for cervical cancer. These could be either chemically removed or surgically removed, because they lead to warts in the genital area. Other STIs are gonorrhea. This is a bacterial infection caused by Neisseria gonorrhea bacteria. It leads to painful urination. On the fetus, it can lead to blindness or stillbirth, which means that the baby is dead as delivered. Treatment by antibiotics, and gonorrhea can also lead to arthritis. It can lead to the PID and infertility. Syphilis is caused by an organism known as Treponema pallidum bacteria. It has different stages, which if you take a pathology course or a bacteriology course, you'll go through these different stages of syphilis. Some of the effects on the fetus, it could lead to miscarriages, premature deliveries, birth defects, or stillbirths. Because it's a bacterial infection, you can treat it by antibiotics. One of the complications of the third stage of syphilis would be dementia. So, early treatment by antibiotics could definitely prevent the dementia from occurring. Trichomoniasis, that is a bacterial infection by Trichomona vaginalis. It leads to itching, discharge. Because the name is vaginalis does not mean it only occurs in females. It can occur in both males and females. In females, if there is a pregnancy, it can lead to preterm delivery and low birth weight. Again, being a bacteria, you can treat it by antibiotics, but there is an increased risk of getting other STIs. And that concludes the reproductive system.